All right. So before I start, I want to emphasize that this project is a really strong community-driven project. So um, I'm just one of the team members. All of the data sets that are released by the Open Scope project are the collective effort of scientists, research associates who you've met actually yesterday, team managers, project managers, engineers that built rigs, as well as animal care, all mice involved, and a lot of leadership was involved in building this platform. So this is really a community-driven effort in many ways. In addition, we are currently funded under the NIH grant till next year. And the project that is selected for the Open Scope program were done so by a panel of a lot of volunteer-driven reviews were done by the community to select which project we run on the platform. And the final decision is made by the Open Scope Steering Committee, which, for instance, XTIMS is part that is listed here. And so I'm incredibly thankful for all of the time that they spend on this project, their support. So you just get at home is that all of the science that is done here is driven by a collective effort. Okay, so what is the goal of Open Scope? Or when Christophe Koch and Clary initiated the Brain Observatory platform here, the vision was eventually to share it as an astronomical observatory. But what this means is that we would run projects not only for the Allen Institute, for scientists across the world. And the process to do that in astronomy is there's like panels of scientists that are reviewing projects on a yearly basis, select the ones that are impactful for the broader community, and run them on the platform. The platform is not only serving the Allen Institute, it's serving the broader neuroscience community. So we wanted to do that. But neuroscience had never done this before. So the reason we wanted to do that is because it will enable scientific discovery, but also democratize and nurture the ecosystem. So that was the goal of Open Scopes. So how do we achieve those goals? We started this effort half a decade ago, and eventually we obtained funding from the NIH to serve all of you guys. So it's an annual cycles of proposals. So every year, typically in the July, we publish a request for proposal call. And then there's a double-blinded review, so which means that the reviewers are blinded to your identity, and you're blinded to the reviewer identity. So science first. So you're judged on scientific merit. And this system is leveraging the pipeline that you saw in the last few days. So many of you have asked, like, this is amazing, but can I use it in my own research? It's just out of my reach. I can't build this. That's not true. You actually can today submit project to this program and eventually leverage our tool. From the beginning, we committed to release the data. So that means that project that is selected, I eventually release to the community. So that's a strong incentive for sharing. And as part of this commitment, we also, in agreement with the NIH, decided to support this workshop. So this is a community effort. Okay, so I'm going to talk a little bit about the project review process with some caveat that I share later on. So as I said, this is science first, so double-blinded. So volunteer scientists across the community are selecting projects. I'm actually not selecting them because in many ways I'm supporting the design because I'm helping people shape projects. So I'm biased, so I cannot select them anymore. It's a two-stage review. So what we set up, and this was the first time we're doing this in the field, so we were just trying to learn from other platforms. So typically in July, we call request for proposals. That will be this month. And then in September, we receive letter of intent, so 2-page long proposals, followed by full proposal in November. So some subset of team will be invited to write a 6-page proposal that will be selected in January by the steering committee. And then over the course of the following year, we will collaboratively work with teams across the world to execute the project within 1 year. But the caveat is this year's is the last year of the grant. And so that means there's no call this summer, unfortunately. But stay tuned, because I think we're working very hard to actually scale up this project, and eventually we can serve many more of you. But in the second half of this talk, I'm going to talk about a community project, which I think you can still engage with. Okay, so this is for context, because we've been doing this for 4 cycles already. And so there's many projects that were selected, that were run, that the science was done, the analysis was done, and then the project was completed. So I'm going to talk a little bit about that. So the process for data release, as I said, is everything goes on Dandy, which is the public archive that Ahad and Carter were alluding to. Typically, the data hands off. So this is the date where the data is finished being collected is about a year and a few months after we meet for the first time to start the project. So that will be this February. And then a year later, we committed for a 1-year embargo. So that means the team that has proposed the project has exclusive access to this data set. In the following year, we would actually make a public data release. So in February, I think this year was March, we actually had an online event, a webinar event where we released those data sets. They are now public, with 2 hours long introductions to the data set. The team presented the experimental design. We explained how to access. There's like notebooks to explain all of that. So this is done with a lot of intent for facilitated data sharing. And here I'm just showing you at the bottom an example overview of the NWB on Dandy. So this is our process, and we put a lot of thoughts into how to document and facilitate reuse. So Carter showed you all of the notebooks, so we actually share code and documentation, so how the experiments with figures, how it's done. So that's our goal for the calls. So what kind of science did we receive? I mean, we've been running this for half a decade now, so you might ask, like, what have you done with that platform? So initially, our platform was really tailored for visual processing. So we had a lot of technologies to record from the visual system. And so we've received a number of projects related to these types of questions. So how does the mouse visual system proceed information? So some projects about how information is shared between the visual system, the visual cortex, and the hippocampus. Some of our projects are looking into how predictive coding might be implemented in the visual system. So that will be, I would say, the general family of family on visual processing. There are also projects that are using the visual system just as a model system to study, for instance, binding. So we had a project on illusion that leveraged a visual illusion to study how two stimuli are related to one another when they form an object. There are projects on temporal coding. And I mentioned, I'm going to talk a little more about predictive processing because this is something that we receive a lot of projects on. And then very recently, we started to touch the pharmacology space. So we actually have done a project on psilocybin this year. So the data actually is collected, and next year we release it to the community. Okay, so I'm just going to briefly go through a few projects that I think are worth highlighting, but they're more than 10 now, so it will go beyond the scope of that presentation. So the first one is a project that was led by Yi-Young Shin. She was initially in Hillel Disney lab in Berkeley, and then moved on to start her own lab in Korea. That study is, as I said, is using illusion as a proxy for studying binding. So when you have 2 objects in the field of view, when they are, sometimes they form an object, sometimes they don't. And so by using illusion, she's using this as a nice trick to really quickly turn it off, the presence of the object. And we've run the project with her. It was a lot of fun. She got access to the data, and then now the paper is going to be in press, I think, in the next month or so in Nature Neuroscience. So it's an examples of like designing the experiments in this way, and then completing the cycle. So we can do this as a community. It's possible. Now, I want to speak a little bit about a few projects on predictive coding. So, and that's going to allow me to bridge. Sorry, I'm just losing battery. Going to allow me to bridge into the community project that we started this year. So this is an older project that we run with Blake Richard, Joel Zilberberg, Colleen Gillon, and many others, where they were interested in understanding how somatic compartment and dendritic compartments encode the presence of oddball and stimuli. So they created visual stimuli that are but had unexpected components, and the theory is that the dendrite and the soma might have different neural correlate as a surprise, and so the intent is to understand how learning occurs across the pyramidal cells. And so we run an experiment with them. They found different responses across chronic sessions between the somas and the dendrites. So essentially, the soma and the dendrite will follow a different trajectory over the course of recordings. So as an examples where we run the project, we eventually it got published, but the predictive coding project kept coming in. More and more people were interested in this type of questions, and so it really felt like, I mean, in some ways, and in the experiments was sort of eerily similar. Like this is like the same kind of questions, so people design the same kind of stimuli, but they're not exactly the same because everybody, you know, they're coming from a different place, and so they will design a slightly different stimuli. But now this is a team from Vanderbilt. Jacob Westerberg was at Vanderbilt at the moment. Now he's in Amsterdam, I think, where they ask us to do neuropixel recording. So great, where we have like a predictive coding experiments with 2 photon, and now we have with neuropixels, also with complementary experiments. But this was designed by different teams, and they found also really interesting stuff with certain types of unexpected stimuli trigger massive response across 50% of the neurons across the entire cortex. But some of the types of unexpected stimuli just recruited a very few set of cells which were actually biased towards higher order nuclei. So it seems to contradict completely the predictive coding framework, which expects oddballs to be more present in lower levels than higher levels. And so already we were starting to see signs of disagreement, even in the studies we were running here. And probably it's related to the experimental design, the differences in experimental design. So, and I'm not going to talk about the 3 more projects on predictive coding that we receive and actually run experiments that are, some of these are actually currently doing analysis on, but they kept coming in. And so at the last year of the grant, and the steering committee responsible for the OpenScope project was really encouraging us to think about how to build synergies across labs, across the community, and to try to encourage people to work together and design experiments together. Because the majority of neuroscience is conducted with like 30,000 independent studies that are eventually colliding each other during the publication stage, which is great. I mean, that's a normal process. But you might argue there's room for some collective experiments that are designed together that are building data sets that are analyzed by a distributed set of teams so that we can answer the question maybe more deeply. And so we thought, given that we had funding already to run experiments, this was like a unique opportunity to try it out. So with the incredible support and help of Michael Berry and Colleen Gillon and Conrad Cording, which is on the steering committee, we tried to make this vision a reality. So we started by calling for a workshop at the Cognitive Computational Neuroscience Conference in MIT in the summer last year. We actually held an event at this workshop exactly a year ago where we discussed the potential of a shared experiments and had a debate with some of you about what it would mean, and started a Google Doc document online with complete free access. So anybody could write anything in that document. with no logging required. With Colleen, Michael Berry, and a few others, Fabian Miklash, and many in the community, we actually started by structuring the document a little bit, so that the contribution would be a little bit structured, and sort of lay out a canvas for this piece. And when we started the workshop at MIT, there was already the start of that document. But after the workshop, it went crazy. More than 50 co-contributors wrote in this document. We had more than 1,900 comments. So like that means you had Martin Dink that disagreed with Andrew Bastos on interpretation of data, and then discussed in public comments about what they think is occurring in those papers. And then students that are watching this can see the current state of the literature, and then understand where predictive coding might be. And so that process has been incredibly fun to me. I learned so much from all of those co-authors. And we eventually ended up with this archive publication, which I hope eventually is going to be published and reviewed, with 50 co-authors, many of which are much more knowledgeable than me on predictive processing. And that document is more than 100-page long. It makes like a review of the field on this particular set of questions. How does the brain predict the next seconds of the world? And it turns out that there's a lot of mechanisms. So before I jump in, I want to spend a little bit of time discussing the science that this review highlighted. But I think there's some lesson learned already. So first, I think scientists want to collaborate in this way. If you just need to set it right, and then they want to do it. I found that making a broad review is a good rallying incentive, because I think it's just it touched on to exactly what a scientist is. We like to understand things and questions the state of the knowledge, and so a review is something that everybody I think can contribute and is interesting to debate. So this was a good approach. We stumbled upon this randomly by chance, but it worked out. It's true that the building shared experimental design needs seed and leadership, so I'm going to allude to that in the next few slides. But a key component to getting this to work was building trust over time. So we met every week on an online forum with everybody, free access. Some people could come in, some people could leave at any time point, but there was a constant flux of information, no secrets. But the process was a learning experience to many. To me, it was certainly is. And the last thing that I think is worth highlighting is that we spent a fair amount of time on the glossary at the beginning of the document, because people disagree what words mean. I still don't understand what adaptation means. I mean, I would like to define it better, but I feel there's like a lot of disagreement. Okay, so generally, I think it's fair to say that the crowdsourcing aspect promoted breadth, because we ended up with a large document, and that the fact that we did all of those interactions online in a written document enable a little more bit of depth, because everybody had time to think about their contribution. Okay. I want to spend a little bit of time describing the review, and this is a long document, so I'm not going to be able to give it justice to it, but you can go, it's on archive, and look at it. What I learned through this process is that predictive processing is an essential functions of probably of what the cortex is doing. Predicting the next second of the world is a really important function. And many studies have tried to understand how this is done by looking at it from a certain angle. So for instance, predictive coding, the traditional models of predictive coding think about hierarchical processing, like how this area contributes relative to with sort of a hierarchy. Errors are going down. That's the idea. But there are many other ways that a system could make predictions, and some of it might not involve computing errors. And so what you see in the literature is that there are models that use different types of computation to make these predictions. that have been proposed. Those are, and we tried to group them: dendritic computation, hierarchical processing, EI balance has been proposed by many models on the way to sort of to make predictions. And a particular one that I like very much is adversarial computation between certain types of inhibitory neurons and pyramidal cells that could be fighting each other, and this way create more complex representation. E to E recurrence has been mentioned in a lot of models. So this is like when a series of pyramidal cells work together to create predictions, and cell type specific computation, as well as stimulus adaptation. And so stimulus adaptation on the bottom right corner is probably the more loosely defined, but generally we think about adaptation about when a stimulus is very, very repetitive and how the system encode that. And some people think about this as like the things that happen in a single cell. Some of them might refer to this like differently, but generally this is the type of computation that people think about. And then when you take the paper in the field, either the theoretical, the experimental studies, they fall somewhere else in that spectra. Like how I'm thinking about dendritic computation done by that is doing some kind of multi-area network computation. Or like it all feels like everybody is trying to understand this process with a single computation. Like for instance, the Rao and Balin model is like they propose one single way that this is done. And I actually find this really unlikely. That's what the cortex is doing. I think it's likely leveraging tons of different computational tricks to do this. And so we have to think about this a little more from a bird's eye view. Like in which context do you use one of these computation or all of them? And the OpenScope project we received, they're doing the same. They were just sampling different parts of that figure. This is the sum of the project we received. So we made that review, and then eventually in the archive paper, there's an experiment that is proposed. And I pushed that quite hard, just to have an experiment design. We had a number of proposals that were, so a number of experiments were proposed by the group, and eventually we had a poll, and we had ask a certain number of questions, which would be, which data set would you analyze first? So if we were to collect this data, would you actually analyze it? That's the really important questions. We actually asked them, would you be interested in contributing data from your lab if we were to do that experiment? And some labs actually said yes, because they felt like this is an important experiment, I'd like to help. So we run this poll, and eventually that allowed us to converge onto one experiment, which is a bit of a combo, but I'd like to explain a little bit. So the idea of this experiment is that there are different types of sensory context for predictions. And so in the case of predictive processing, if you actually look at the literature, there are very many labs use drifting gratings to create different kind of sensory context. And they will play with them in slightly different ways. So some of them will have like a sensory motor coupling system. So the gratings flow, and it's controlled by the movement of the mouse. So now you think, how does the mouse predict the next grading? It has to integrate its own movements, and then some sense of time, and then some sense of what's on the screen. Some of the stimuli, like the local global oddball experiments, you have a succession of drifting gratings. They're not coupled to the mouse movement. So now you're probably talking to a very different network. And then the local oddball is very easy to predict if you just think about the last seconds, whereas the global oddball, you need to remember at least 5 seconds worth of data. And so it's likely that in one case, you might be able to do this with just local recurrence. In the other case, you might actually have to recruit the prefrontal cortex and then in V1 listen to that input, depending on how much sort of depth you need. These are just hypotheses at the moment, but what we are trying to do here is run 4 types of predictive processing experiments that have actually been published, and then run them sequentially. So in the same mouse, we'll record all 4 types with either 2 photon or neuropixel recordings. So this way, we hope that a multimodal data will allow us to sort of test this different computation and then also compare the responses in the same mouse. So the intent is to study whether actually this network are very differently engaged in all of those tasks and generate a data set that can be used for modeling across the different types of audible. So that's the next experiment we converge onto. And the review was finished in February, approximately this year. And we have already started experiments. And we are doing this extremely publicly. So contrary to most, I mean, a neuroscience project these days is you have a great idea and you don't talk about anyone about it until you're like fairly ahead in your analysis and you're confident about your results. And so we're just sharing day 0. Like we shared day -5 when the experiment was not even designed. So we shared the process of doing the review, the process of designing the experiments, the process of collecting the experiments, and the data. We have a project website that highlight everything that I've talked to you about. So you can go there, you can see who is engaged in that process. And when we meet every week on Tuesday mornings at 9 a.m. on YouTube. And so any one of you can join the discussion. I mean, so far it's been 20 to 30 people every Tuesday join. And we also have a lot of discussion on the GitHub discussion forum. We exchange analysis. So it's a really a strong experiment in social neuroscience, I would say. In the last few months, we actually collected a little bit of dendritic input data on the SLAP2 platform that Kasper Podgorski and his lab have. We have some amazing pilot already results on this that show when you change the sensory context, the strong changes in the input to the pyramidal cells. So some of the people that are analyzing this data, I think, are really excited to see where this is going. I'm certainly looking forward to what's going to happen in that next year. So data collection is ongoing. As I said, we have some SLAP2 data, and we're going to have more neuropixels and mesoscope data coming out over the year, and we'll push it out as it comes in. And anyone can join. So if you want to do some fun analysis without having to bother, you can just join us. That's it.